Hi, my name is Chi-Chung Li. I currently oversee the immune bispecific antibodies portfolio at Genentech Roche in the Department of Clinical Pharmacology. Today I'll be sharing with you highlights from the MIDD experience for Mosunetuzumab. At the heart of this work, I believe that we share the same mission of making smart and science-informed decisions for the benefit of patients. The MIDD pilot really helped promote proactive dialogues with the FDA to enable that. Mosunetuzumab, or Mosun in short, is a bispecific antibody that targets CD3 on T cells and CD20 on B cells in development for the treatment of B-cell malignancies. It is a full-length humanized IgG1 antibody, so it has a longer half-life compared to fragment-based bispecific antibodies, which requires constant infusion of drugs. Mosun instead can be given conveniently once weekly to every 3 weeks. The beauty of bispecific engagers is the ability to achieve durable anti-tumor efficacy through simply the simultaneous binding to T cells and the tumor cells, which creates an artificial immune synapse that activates the T cells, leading to release of cytotoxic granules and killing of tumor. This mechanism of action bypasses the typical physiological requirements, which involve antigen presentation, upregulation of co-stimulatory molecules, and in the proper context of cytokine release. Notably, as an conditional agonist, the efficacy of Mosun can be achieved at subsaturating levels of target receptor occupancy. A key challenge in drug development, however, is cytokine release syndrome, or CRS. It is a major safety concern across T-cell engaging bispecifics and also CAR T-cells. CRS is an untargeted effect. The T-cell activation as a result of target engagement leads to a cascade of immune activation, resulting in systemic cytokine release leading to CRS. In principle, pharmacologically, one would expect the higher the dose, the higher the target engagement, leading to higher safety risks. As such, a key task in phase 1 development for these T-cell bispecifics is to figure out a way to mitigate CRS so that we can get to an effective dose level. To understand CRS, it's important to understand IL-6 and its kinetics. We know that patients with CRS tend to have higher IL-6 on average, so it is a useful biomarker to draw inferences to CRS risks. This was done for Mosun through a quantitative systems pharmacology model. Briefly, this model describes the kinetics and trafficking of both B cells and T cells in several physiological compartments, including the blood, bone marrow, spleen, lymph nodes, and tumor. It describes the B cell generation, maturation from the bone marrow, exit and traffic into other tissues. It also describes T cell generation from the thymus into the blood, and T cells can also traffic into different tissues. In all compartments, the T cell activation is described as a function of the concentration of Mosun in the T cells and B cells, and these are the upstream driver for IL-6 release. And this model was leveraged to inform the phase 1 dose escalation design for Mosun. As you can see on the right, the model is used to predict and compare 2 different dosing strategies and their resulting IL-6 levels. The model predicts that if one gives a small dose, 1.6 milligram in this illustration, that precedes the target dose, or 20 milligram in this illustration, by so-called step-up dosing strategy, it leads to a much lower IL-6 peak predicted in patients than if one were to administer the target dose directly. In other words, the model predicts that you can get to the same target dose level, but with only half the IL-6 through step-up dosing strategy. And the lowering of the IL-6 is expected to reduce CRS risks. The model also predicts an important kinetic feature of IL-6, where it is primarily a first dose effect, and repeat dosing seems to result in minimal modulation later on. Pharmacologically, the postulated mechanistic explanation is illustrated here. The target engagement by Mosun results in T-cell activation both in the peripheral blood and in the tissue. The peripheral activation contributes directly to the cytokine release in the blood compared to the tissue activation, which is to a smaller extent due to tissue to blood partition. Upon repeat dosing, since the targets are depleted by the drug, there are few targets left that are immediately accessible from the blood, leading to lower IL-6 with repeat dosing. Here's the actual clinical data following step-up dosing, which is consistent with prediction. On the left shows the PK, represented by mean serum concentration of Mosun over time, colored by dose groups. As expected, PK accumulates and increases with each escalating dose level of the step-up dosing regimen. In contrast, on the right, IL-6 concentrations peak primarily following the first step dose and remain low upon repeated dosing despite the escalating dose levels. We are again seeing the unique IL-6 kinetics that change with time and diminishes upon repeat dosing. In terms of clinical CRS, the data was consistent as well with expectation. CRS was primarily a first cycle effect, consistent with that of the IL-6 time course. On this slide, we're looking at the rate of CRS on the y-axis in terms of a probability of a patient experiencing grade 2 or higher CRS versus Mosun exposure modeled by peak target engagement level on the x-axis. We're comparing two dosing regimens, a fixed dosing regimen on the left versus a step-up dosing regimen on the right. The green dots are patient-level data. 0 means no event and 1 means yes. The blue trend line is based on logistic regression, where the numbers represent the observed CRS rate in each tertile of exposure. Overall, we can see in the fixed dosing group that there is a trend of increasing CRS rate with increasing exposure. However, when switched to the step-up dosing on the right, despite going to greater than 20-fold higher dose levels, the CRS rate did not increase significantly, indicating the CRS mitigating effect through the use of step-up dosing regimen. Essentially, the step-up dosing shifts the exposure response curve to the right, via which the therapeutic index was expanded. Notably, the CRS was mild and transient. No MTD was reached across 3 orders of magnitude of dose ranging. I want to briefly mention that there is an additional mechanistic explanation at play that has to do with desensitization of T cells to release cytokines such as TNF-alpha upon repeated dosing, leading to less activation of macrophages and monocytes to produce IL-6 upon repeat dosing. More details can be found in this paper by Li et al. Listed here. Importantly, this desensitization does not impact tumor killing capability. In other words, we start to decouple the drivers for safety from that for efficacy upon repeat dosing. Now, switching gear to efficacy. When we first looked at the clinical dose response in patients with aggressive NHL, we were very puzzled. Here I'm plotting the complete response rate in orange and the overall response rate in purple, summarized across dose levels. Despite having a robust 500-fold dose ranging in the clinic, it's apparent that there was no dose response. And the usual caveats of small sample size alone doesn't fully explain it, as even with further analysis pulled across dose groups using PK, it still indicated no obvious dose response. So we took a step back and realized what could be going on is a confounding from prior treatment of anti-CD20 therapies at the target engagement level. So this needs to be addressed in order to characterize the intrinsic dose response. In a late-line relapse refractory setting, all of our patients received prior anti-CD20 therapies such as rituximab. The residual levels of rituximab in our patients bind to overlapping epitopes on CD20 as Mosun does, and competes for target engagement transiently and significantly confounded the dose or exposure response relationships. What's going on at the target engagement level is illustrated here. Mosun works by binding to CD20 and CD3, which creates an immune synapse that drives anti-tumor activity. The residual levels of prior anti-CD20 therapies transiently competes with Mosun on its target engagement and reduces its anti-tumor effect. And of course, this is reversible at higher doses of Mosun. Knowing this pharmacology allowed us to use mathematical ways to account for the transient competition of rituximab through the use of a model-derived receptor occupancy level in order to address this confounding appropriately. Based on principles of binding kinetics at equilibrium, one can derive the receptor occupancy percentage based on the PK of Mosun rituximab as well as their respective binding affinities. So using this model-derived receptor occupancy instead of dose or PK, voila, we got beautiful exposure-response relationships for complete response rate and overall response rate. And this is exactly what's needed to guide the proper dose selection in order to maximize clinical benefit. And importantly, this also revealed the true therapeutic potential of Mosun if dosed properly, which otherwise would have been masked had one assumed no dose response or stopped at a lower dose limited by adverse events. So to put it all together, Mosun works by engaging the T cells and the tumor cells to create an immune synapse to activate the T cells, which drives tumor killing. It also leads to a cascade of immune response to produce IL-6, leading to CRS. The integrated picture of the therapeutic window can then be represented by exposure response curves. For efficacy, we were able to characterize the exposure response by addressing the confounding effect through modeling of the receptor occupancy and saw an increasing efficacy with exposure that eventually plateaus at high exposure levels as shown in blue. And there is also an exposure safety relationship that reduces with repeat dosing as shown in red. Step-up dosing works by keeping the first dose low on the exposure safety curve where it's most potent, and then escalating the target doses later to maximize efficacy with a reduced potency for CRS. In summary, the key learnings from this experience: the model-based dose selection strategy led to enhanced therapeutic index and science-guided dose escalation to maximize the therapeutic potential and accelerated the development of Mosun. Modeling was used effectively as an integral part of Mosun clinical development, which led to the use of novel dosing strategy for a new modality and the use of novel endpoints to guide decisions. And we didn't have time to talk about this, but this modeling also provided insights to inform development across NHL histologies. I'll end with the acknowledgement slide. My email is provided here, and I'd be happy to engage further. And thank you so much for your attention.